Hi, biology, Mrs. Cozen. This will be video 5, multiple alleles. We get to use 3 letters now. Yay! We're going to get to see some more genetic variety now. Up until this point, you guys have only been using 2 letters. Maybe you had a red and a white, and the red was dominant and the white was recessive, and you used an upper and lower case. Maybe you used 2 uppercases and made like black and white for codominance, and black and white made black and white stripes. Now we're going to add an extra letter, which gives us some more variety in our phenotypes. The one that I'm going to show you, if you guys will look at this right here, kind of read this over, what do you think my example is going to be about? If you guess blood types, you're right. It's going to be about blood types. I'm going to teach you guys how to determine what kind of blood types you would see in an offspring based on the blood types of the parents. We are not going to touch on the whole like O positive, O negative yet. That negative is a different gene. We'll talk about that a little bit later. We're just going to talk about how do you get type A blood? How do you get someone with type B? How do you get someone with type AB? And how do you get someone with type O? The first part of this right here, these are phenotypes. So please label this as phenotypes, okay? Physically, you have type A blood. The second part right here, please label this as genotypes. These are the different genotypes that will give you this phenotype. Type A, you could have homozygous type A, where you've got 2 of these dominant alleles, or you could have heterozygous type A with a dominant A and a recessive O that it's hiding. That is the way that part of this is underneath complete dominance. Type B works the same way. You could have 2 uppercase B's, homozygous type B, an upper and a lower, heterozygous type B. Now, if you have type AB blood, awesome, you have one of the rarest blood types, but also your alleles work together like codominance. A isn't dominant over B or B isn't dominant over A. They work together, just like in the black and white example I said earlier, like black and white would work together to make black and white stripes. And then the last one, type O, is recessive. Most of you guys probably have type O. The majority of the population has type O blood. If you don't know what type of blood you have, go home and ask your parents, see if they know. If you ever to give blood to the Red Cross, they'll tell you as well. Now, your Punnett square is going to be a little bit bigger because remember, we have a much larger genetic variety here because we get to have 3 alleles. Yay! That is why I am not in the video, because I wanted to make sure that my picture of me and my ugly dog don't get in the way of you getting all the genotypes and phenotypes you need. So starting here, we've got one parent. This parent has type A blood. They are heterozygous type A. Dominant A, hiding a recessive O. This parent has heterozygous type B. Dominant B, recessive O. Filling in our Punnett square here, you get 1 when you get a allele, from this parent and a B allele from this parent. So this possibility is AB blood. They work together like codominants. Anytime that you have an A and a B and an O, though, or an A or a B with an O, it works like complete dominance. The O is masked. The O gets bullied by that dominant gene. So this possibility is type B blood, heterozygous. This is type A blood, heterozygous. And this is homozygous type O blood. Really, you can only have homozygous type O blood. There's no heterozygous type O blood. If you had a heterozygous, it would end up as this type B or this type A. So there's really only homozygous type O blood. Now over here, lots more genotypes, right? We only had 3 genotypes before, and now we have 6 possibilities. We've got 2 that will give us type A, homozygous type A and heterozygous type A. I did not have any possibilities that were 2 uppercase A's, so that's why that one's a 0. Make sure you write all of this, even the stuff that's in the parentheses over here. We do, however, have 1 possibility in our offspring of a heterozygous type A. That's why this one is 25%. We have no offspring possibilities that would be uppercase B's, homozygous type B, but we do have 1 that's a heterozygous type B. And then we have 25%. One of our possibilities is AB blood, heterozygous AB. And then we have 1 possibility of type O. So you guys can see we've doubled our genotypes just by adding 1 more letter. That's a lot to do. And phenotypes, now instead of 3 phenotypes, we get 4 of them. Again, write all of this. I want you guys to write everything, even the stuff that's in parentheses. I added the stuff that's in parentheses are the genotypes that go with the phenotype. So we go at 4 possibilities here. Type A, type B, type AB, and type O. Only one of our offspring was even possibly a type A. You would add together all offspring that are homozygous type A and heterozygous type A. And that gives you that percentage. Type B, you've got homozygous type B and heterozygous type B. You add those together to give your percentage here. AB, it's only one possibility to have type AB blood and one to have type O. So this cross of these 2 heterozygous parents gives us a huge variety in blood types of our offspring. Make sure you guys have all this stuff written down before you move on. And here's your summary. Make sure you write the question, guys. You've got to write the question. If you get any part of the Punnett square wrong when I'm going over it with you, it's tough for me to pick out what you did wrong if I don't have the question to kind of underline vocabulary for you. So make sure you write down the question for me, okay? Then you guys need to do Punnett square. You need to do all the possible genotypes, all the phenotypes. So you guys need to do what I did on the slide before this, but it needs to be an AB mother and a heterozygous B father. Remember, hetero means different, okay? That is the end of video 5. Good luck with your blood typing.